Good morning, everyone, and welcome to our first session for OmicsLogic Transcriptomics. Today in this session, we will review the program, make sure that everybody is set up and ready to go. And for those of you that are joining us for the first time, you'll get an introduction to what transcriptomics is all about. OmicsLogic Transcriptomics is an online training program for students, researchers, and clinicians. It is designed to introduce participants to the various aspects of RNA-Seq data analysis in application to basic research, biotechnology, and biomedical projects. And you'll see that we'll talk today about RNA-Seq in application to cancer research, infectious diseases, and biotechnology. But we'll take a perspective from the data point of view. So, OmicsLogic programs were designed in collaboration between Pine Biotech, which is a big data analytics company based in the US, and numerous academic institutions that have participated in content evaluation, curriculum review, and project design. So as a result, the OmicsLogic programs have been thoroughly tested by both faculty and students, and we now know that they lead to excellent outcomes for students and faculty that are interested in bioinformatics, data science, and biomedical research. One of our primary collaborating institutions is the University of Haifa, and primarily the Tower Bioinformatics Research Center, but also some of the other institutions that we've worked with are both in the US and around the world. For those joining us for the first time, my name is Ilya Brodsky. I'm the co-founder and CEO of Pine Biotech. Our vision is to enhance human health and well-being by enabling biological research and discovery with relevant data, solutions, and support. Our mission is to simplify bioinformatics and advance research through our modular and intuitive multi-omics data analysis platform that is powered by human experience and artificial intelligence. We offer training, research support, and big data analytics tools designed for research and biomedical discovery that does not require technical expertise. Our mission is to support cutting-edge research and solve big data challenges by making our solutions easy to use and train those interested to use them to arrive at sound and reproducible results. I'm honored to have been working with people that have much more experience in this area than myself, including the other co-founders of the company, Dr. Alfred Tauber, who comes from a medical background with expertise in oncology, immunology, and biochemistry of inflammation. He has a long-standing recognition for his generous support of many international projects and research activities. One of these projects is the establishment of the Tower Bioinformatics Research Center, which has been at the core of what we do and a close collaborator in numerous research projects. Dr. Leonid Brodsky is the director of the Tower Bioinformatics Research Center and is an expert in bioinformatics and biostatistics. He has numerous publications on bioinformatics, and over the course of his career has developed multiple novel algorithmic approaches to biomedical and basic biological data research. These have been widely used in basic and translational research projects in biotechnology, pharmaceutical R&D, agriculture, and biomedical research. What we will discuss today is also a result of hard work by some of the people that are mentioned here on the left. Julia Panov, who is an expert bioinformatician and has helped prepare many of the materials that we will utilize. Avi Titievsky, who is a computer scientist and leads the development of the T-BioInfo platform that we will use. Mohit Mazumdar, who is in charge of business development for Pine Biotech. Joshua McKendall, who maintains the educational web portal that we will leverage. And Bipsa Biswas, our online community manager and support specialist, who I'm sure that many of you have already interacted with and will continue to interact with throughout this program. I also want to introduce you to Dr. Harpreet Kaur. Together with me, she will be co-hosting some of the sessions, and she is a certified OmicsLogic trainer, which means that she has completed a PhD in bioinformatics, but also completed all of the training resources that we will utilize and has helped develop some of them to make them adaptable to these online training sessions. As I mentioned, she has a PhD in bioinformatics, and her focus was on cancer genomics, and she has a lot of experience working with transcriptomic data. She will help conduct some of the sessions and work with each one of you as a mentor and a support specialist. She can answer questions about any of the pipelines that we will run. If you face any issues with files, settings, or formats, she is also an experienced researcher herself with many peer-reviewed publications that she contributed to or has led independently. So if any one of you has research questions, you're also welcome to set up a time to discuss with her, and we'll go through those technical details in the in the following slides. So before we continue, I wanted to make sure that all of you that are here today have completed setting up your accounts. I see that we have quite a number of you here on this session. So let's go together to edu.t-bio.info. This is a website where we will add different types of materials for you to go through. So please go with me to edu.t-bio.info. And please let me know once you are here. You can simply post in the chat that you're there, or yes, or something like that, so I will know. Okay, so once you're here, if you have already completed your registration, you should be able to see that you are logged in, just like you see right here. For me, I can see my profile. And I will use for a second Dr. Harpreet's profile as an example of a completed profile. Okay, so if you click on the right corner and you get to your profile, you will see that your profile has an avatar. Doesn't have to be your personal picture, but it can be some kind of an avatar so we can easily find you. You can also add some social media links to your profile. This is good for communicating with others that are also going to be participating in this program. You can even add your research accounts here as well. And if you would like, you can add a little bit of information about you. So as you can see right here, Dr. Kaur has added some information about her research background and interests. And she also has a faculty position, which she is an OmicsLogic Bioinformatics trainer. Now, what will happen throughout this program is on this account, you will see how all of your activity is going to accumulate to demonstrate your progress. This means that any courses that you will go through, you will get certificates for. Any exercises that we will do, you will also see them appear here under activity. These will include pipelines that you run and coding exercises that you complete. So please make sure that you finalize your account. You can do it during this session or after this session. So you can see here, for example, Dr. Kaur has completed Introduction to Metagenomics or has run specific pipelines. This will help you keep track of where you are and also will help us evaluate whether we are going through the material too fast or too slow and make sure that everybody is staying on track. Now, to get access to all of the resources for this program, if you have completed the registration, you will have access to this organization page. So this organization page is the page where we will centralize all of the resources associated with this program. The way to get there, an easy way to get there, and remember this link, is just by going over to events, and you will see that this is the first program that appears right here on the left, OmicsLogic Transcriptomics. So if you click there, you will get to this program, and what you will see is the schedule of upcoming sessions, as well as the updates from all the participants. For example, pipelines that have been run or coursework that has been completed, and exercises in coding that have been completed. So again, we will review these in the interactive sessions, but you will also see all of that come up here. Also, this program page has a forum. And this forum includes, okay, let's go to the forum. This forum includes important announcements that will have reference publications for the material that we will review in the sessions. It will have session recordings, as well as any technical questions. So the most efficient way to communicate with us, and especially if it is relevant for the rest of the participants, the best way to talk about these is by posting a forum question right here. If you have trouble with a specific course or a specific pipeline or a specific assignment, you can also reference those by pasting your URL, an image, or a video so that we can clearly see what the problem is. So in this program, we will see how relying on analytical methods and reference databases, researchers can study RNA-Seq data to understand how gene expression variation happens across many conditions. To do so effectively, bioinformaticians leverage big data processing methods organized into bioinformatics pipelines, perform exploratory data analysis and normalization, and then apply statistical methods for rigorous hypothesis testing. And this is what's called standard RNA-Seq. So what we will hope to cover in this program are methods for data processing, exploration, statistical analysis, and we'll also touch on machine learning because we are moving into the age of big data where large biomedical research data sets are available for mining and classification. At the end of the program, we will also speak about single-cell RNA-Seq, which is a hot topic and an important one as we try to understand how disease is manifested in different tissues, organs, and cells. But before we jump into all of these specific types of sessions that we have scheduled, let's do a little overview of what next-generation sequencing can help us understand and how does that correlate with the biology of what we are studying. Omics technologies like next-generation sequencing can be used to explore the roles, relationships, and actions of the various types of molecules that make up the cells of an organism. Many types of omics data can be generated using next-generation sequencing, or what is also known as high-throughput sequencing. The data can show detailed information about genomic variants and patterns of gene and isoform expression. So when we think about a cell, we are thinking typically about its phenotype. And collectively, different ways to categorize and characterize phenotypic properties can be referred to as phenomics. Here you can see a microscope image of cells that reveals cell morphology and different physiological properties that help us characterize this specific type of a cell line. But this type of cell line, this phenotype, is based on how the processing of DNA happens. And so when we talk about genomics, we are talking about the genetic code that encodes specific genes and coding sequences that regulate how this phenotype appears. Along the way, from the genotype to phenotype, we encounter what's called epigenetic regulation. That is regulation that affects gene expression, but is not necessarily encoded in the DNA. And then we go to transcriptomics, and that is really the study of how to quantify different types of coding sequences from the DNA that are being transcribed or expressed. And that will help us estimate approximately what kinds of proteins are being produced. But the proteins themselves are not exactly what is encoded in the DNA. So it is a proxy for expression of proteins, and we will primarily focus on the aspect of protein coding mRNAs or messenger RNAs. So as a method of measuring RNA translation, transcriptomic studies can help detect mRNA expression inside the cell as an accurate alternative to membrane protein expression detection, which allows for molecular precision in diagnostics and even development of treatment based on molecular pathways. So when we speak about transcriptomic or transcription data, we're talking about the process of mRNA being in the act of translation, which helps us estimate the resulting protein expression. Now, in biomedical studies, this is a useful alternative to measuring protein abundances. And so we will see how that is used in many different types of biomedical applications. RNA sequencing, or RNA-Seq, has revolutionized the way biologists examine transcriptomes and has been successfully applied in biological research, drug discovery, and clinical diagnostic development. Compared with microarray-based transcriptome profiling, RNA-Seq has a wider dynamic range, which means that it can detect a wider type of variation and avoid some of the technical limitations such as varying probe performance and cross-hybridization. RNA-Seq can measure the expression levels of thousands of genes simultaneously and provide insights into functional pathways and the regulatory networks in biological systems. In addition, RNA-Seq can provide novel insights into alternative splicing, unannotated exons, and novel transcripts. Most importantly, such detail has already proven to yield significant discoveries in many research areas. So let me pause here, and I wanted to ask a few questions about your personal background. So you might be coming from a biological research background, from clinical research background, maybe you're in industry, or perhaps you're a student and you're still undecided about where you're going. So can we take a few minutes and talk a little bit about what is your background? So you can, for example, put in, I'm a master's student and I'm studying, let's say, biomedical informatics or something like that. And the other question that I have is really where you are from. So you can mention your country, your city and country, or even the continent that you are from. And you can type your responses in the chat box. So I'll put mine in. All right, thank you, Andrea. Clinical research, statistical development, applied research, PhD in clinical community psychology with emphasis in quantitative psychology, USC. Okay, bachelor's degree student studying medical laboratory science in Nigeria. Aisha is a medical laboratory science student, undergraduate. Hebrew is a bachelor's degree biochemistry and molecular biology in Kenya. Raju is a master's computer science student in India. David is a postdoc at the Massachusetts General Hospital in Boston, biomedical research on autoimmune diseases. Okay, great. Aisha is a medical laboratory science student from Nigeria and Africa. Nanpun Singh is a lecturer in medical technology in Thailand. MHC18 is a bachelor's in molecular biology from University of South Florida, taking a course because of interest in genomics and epigenetics. And Marek is a co-working in research biotechnology. All right, great. So thank you for sharing that information. Devu, cancer researcher, master's graduate in biology from Kerala, India. Monojit is a master's in zoology fishery science from Kolkata, India. Felicia is a microbiology lecturer from Nigeria, Africa. Okay, excellent. So we have here a lot of different locations. Francisco Ferreira is a veterinary scientist with PhD in parasitology from Brazil, working with vector-borne diseases at Rutgers University in USA. Krupa Ann Matthew is a PhD degree holder in biotechnology from Trivandrum, India. Excellent. So we have many different locations. It's exciting to see all of you joining today. So let's talk a little bit about NG is also a PhD in bioinformatics from the UK. Great. What type of applications are there for bioinformatics in these different applications? Really what we will focus on in this program is how can gene expression and the patterns contained in such data can be associated with clinical phenotypes or clinical data and help us identify molecular biomarkers or specific types of patterns that can be useful for diagnosing disease, selecting therapies for different patients, and even identifying targets for specific types of treatments. So the type of data is really a view of a particular type of biological phenomena. But the way to use this specific data and the patterns contained in the data is driven by the application. So as we discuss these different types of data, we should consider that it has applications in many different domains. And that's why one of the ways that we will apply RNA-Seq analysis approaches is to look at specific projects. And so as we look at these projects, please keep in mind that agriculture, neuroscience, cancer, other types of applications are all going to be related to whatever project you can find. And the easiest way to find a project and start thinking about the applications of RNA-Seq in the area of your interest is to start exploring publicly available data. So one of the important things that we'll do in this program is we will introduce you not only to the theory and the tools for RNA-Seq data analysis, but we will also go and find different types of data sets that you can start analyzing. So some of these projects that we will explore include diagnostics and cancer biology. In specific, we'll talk about the relationship between tumor and the tumor microenvironment. We'll talk about different types of clinical phenotypes of different types of cancers. We'll also talk about disease classification and patient stratification, which is a way to define disease not based on clinical observations, but really based on molecular manifestations of those diseases, which also helps us understand variability in the patient population. And we'll also talk about single cell and tissue composition because we will explore how disease is manifested in different tissues in different ways. And it's important for us to really link those molecular patterns that we discover at the individual cell type level. So to summarize, in this program, we will focus on specific topics that will combine the logic of analysis with some practical hands-on assignments and repeating those assignments in sessions like these that will allow you to understand and practice methods of analysis and interpretation of RNA-Seq data and use them in real-world examples that we will find in publicly available data sets. So at the end of this program, you should be able to plan how to go from raw data, and we'll talk about several different formats of the data that we start from, then go through the steps of processing and exploring that data, and then understanding how to identify relationships between what we find in the data from sequencing and some phenotype that will lead to biological interpretation. And we will be able to plan how to apply such methods to a project of your own, knowing where to find specific types of data, and then plan our bioinformatic analysis. So before we move on, let's make sure that we understand the schedule. So to understand the schedule of this program, for those of you that are registered, we already looked at the specific program page, which was right here, and you can look on the home page to find all of the sessions. But we can also look at the schedule right here at the registration page. So for those of you that have not yet completed your registration, I recommend going to this link, okay? And Andrea is asking, are the slides for each workshop available for note-taking? So what will happen after each session is we post the video, and it will be found right here on the forum. You will have a special section that will say session recording, so all the videos will be posted there. Now, a lot of the material that we will talk through is also found in specific courses, and I will get to that in just a second. Okay, so for those of you that are coming to this program just to explore right now, I just wanted to point your attention. Right here, you have the specific sessions that are being planned and the dates and what they will cover. So please review this schedule, and based on the schedule, you can also find some of the resources that come together with the program, because the sessions that we have are just the glue that kind of links all the different elements together. But we have a lot of other things that we have prepared for you to look through. One of the important things that are included are online asynchronous courses that you have access to as a part of this program, and some starter projects. So these are projects that are based on published peer-reviewed publications, and they allow you to see how the RNA-Seq data is used in all kinds of different applications. You will also get access to a big data analysis platform that allows you to quickly process data and have access to multiple different types of algorithms that are somewhat cumbersome to install on your own machine, and definitely time-consuming to run. So it will give you practical experience working with these different types of algorithms without having to have huge resources at your disposal and really a complicated tutorial to install all of those. We will also practice some of the analysis methods on a coding platform that we developed that will allow us to do it together during these interactive sessions. Okay, so for any of you that are having questions about registration or any kinds of payment options or others, please email to marketing@pine.bio. I'm just seeing some messages here in the chat, so I'm just going to put this marketing@pine.bio. So you can email any of your questions about registration or access to any of these tools to marketing@pine.bio. Or Bipsa, thank you Bipsa for posting your email here as well. You can reach out to her on chat privately to also discuss those options as well. So as I mentioned, on this page, you will find all the details related to registration, access to different resources, as well as the schedule. And once you have completed, you will have access to this program page. On this program page, we will post any kinds of additional reference materials for you to go through. Because as we go through some of the resources in the program, there will be additional reading materials like publications that review the methods that we discuss. There will be projects that we will go through, and we'll post links to those project datasets. And so all of that will be right here on this program page. At the end of the program, you will see that your profile is going to start to improve, and you will start seeing points for different types of activities that you perform. For example, after completing a pipeline, after going through quizzes, after going through a course, you will start seeing that you have an activity tab with the total number of courses, with the pipelines that you've run, and importantly, for each one of the sessions, for each one of the asynchronous courses that we will go through in this program, you will get an independent certificate. And after completing the whole program, you will also have the ability to get a certificate of participation or a certificate of excellence. The certificate of excellence is really for those that have done all of the expected assignments, but also have gone beyond. So really, to quantify that effort, we've put a number to it, and it's about 10,000 points. So as you see your points accumulating, you will know that after you've crossed 10,000 points, that's when you get to a certificate of excellence. Now, the program, as I mentioned, is composed of these several different elements. They are interactive Zoom sessions like we are doing today. They include a lot of self-guided study materials, and those self-guided study materials will be available on the educational platform as well as the OmicsLogic platform. I'll go through those in a second. But inside of those, there will be some very practical assignments that include running pipelines and exploring the outputs of those pipelines, and also some coding exercises. The coding exercises are not a requirement, and so we will try to show you how to easily apply some of the methods on the platform first without any technical requirements for coding. But then if you want to understand how those methods work, you can actually open up that code and see what does the code say and how to modify it so that it works on your data, producing the results that you can work with. So before we go on, I wanted to make sure that I'm addressing any of the questions that are starting to appear in your mind. So if you have any questions, again, the best way to post them is to put them in the chat. As you post them, I will start addressing them at specific breaks that I'll take throughout this presentation. And so in the future presentation, the same process will happen. We will first address some of the questions that you have posted on the forum or by email, if they are relevant for everybody in the session. Then we will review the material that we have covered in online courses, and then we will do some practical exercises together. So again, if you have any questions as I am speaking, you can post them in the chat box, and I will address them as we go through the slides. So how is this online coursework organized? The transcriptomics program is really covering the material that is found in transcriptomics 1, 2, 3, and 4. And this is a collection of courses that is an increasing progression of difficulty, but also what's called downstream processing steps. When you start with the data, it has to be generated. And so the generation of data, what that data represents, how that data is processed, so how to get it from its original digital format into a table of gene expression, is covered in Transcriptomics 1. Then we will talk about statistical analysis. So that statistical analysis helps us understand the specific patterns and how they might be associated with phenotypic information we have about our samples. So we'll talk about differential gene expression, factor analysis, and how to use regression to evaluate associations between gene expression profiles and the factors in our experiment design. In Transcriptomics 3, we will talk about what's called big data analysis. And big data analysis opens up the opportunity for application of data mining and classification to gene expression data. Gene expression data is what's called continuous data, and we will look at how to explore that type of data for specific patterns using principal component analysis and clustering, but also how to train a machine learning model to develop what's called predictive analysis using classification. In Transcriptomics 4, we will talk about single-cell transcriptomics or single-cell RNA-Seq, where we will talk about how to apply some of the similar methods, but in a different way, to the problem of zero-inflated data, which comes from thousands of cells that are sequenced individually. Within these courses, you will find a video introduction, and you will also find a curriculum that helps guide you as you go through the material. So for example, you will find in Transcriptomics 1, a basic explanation of transcription and translation, but also will provide you some controls. And so some of you were asking about notes. Here in the top menu, you will find some of the resources that you can take with you. So this will include a course outline, you can search for specific sections, and you can also access a lesson forum. So each one of these lessons has a forum, and also download the PDF. So if you want to take these materials with you, you can download the lesson PDF and go through that when you are offline. Now, the course materials are available as a downloadable PDF, but that is going to be limited because in the actual course material, you will find specific terminology. So terms that are important as you go through the full sequence of this training, terms that are important for you to remember. You will find them underlined like this, and you can hover over to get a definition of that term. So it will help you understand all of the material without having to look up those terms. Inside all of these courses, there are practical assignments. And so these practical assignments will help you understand what we are talking about in a practical way, how to apply these methods to data sets. So here, for example, you can see a hands-on assignment for Transcriptomics 1. You will have some time to go through this assignment. So once you are starting, that time is going to start. And then you have a passing grade. So there will be some very specific types of assignments here. And those assignments will include some running of pipelines on the T-BioInfo platform. So let me show you how this platform works. So to see how this platform works, you need to go to server.t-bio.info. And for those of you that have completed the registration, you should be able to access this account using a password and an email address that was emailed to you. So here I know that I am logged in. I can hover over this icon right here and see that my name is right there. And once you are in, you will have access to different sections of this platform. So please let me know how many of you have found their way onto this platform already. Maybe you can put 1 for those of you that are here and are logged in, and 2 if you are having trouble getting access to this platform, but you have requested access. So if you don't have an account, you don't know how to log in, you can go right here to access and fill out this form. When you fill out this form, we receive this notification and we have to give you access based on your request. Okay, so David has trouble accessing. So Beepsa, if you could please look into that. Okay, David, it looks like you need to check your email. Okay, great. So for those of you that have access, you should be able to follow the steps that I'm going to do here and do them together with me. So the way to do this, and we'll just do a demo here real quick to just get a sense of how this works. Let's go, you can see under NGS data, which is next generation sequencing data, you can see the section for transcriptomics, and this first section is called RNA-Seq chip parallel analysis of NGS and microarray data. And do not click on this drop-down arrow, and you should see a number of demo pipelines that pop up. Okay, and here we will go through this pipeline right here, RNA-Seq cell line project. Once you're on this page, you can start clicking on buttons that are lit up, that are dark and have this yellow border around them. So this is what we will go through in this program. We'll go through the steps of processing, mapping, and quantification of gene expression levels. And here you have an example pipeline that we will try to replicate today, just so that we understand how this will work. So the first logical step is pre-processing. Essentially, we are taking the reads that have different main sequences that come from the genetic material in our sample, and some technical sequences that include adapters, and some of them will have all kinds of other technical sequences. So to process these reads, we need to eliminate all of these technical sequences and only leave the valid sequences. And so this is done by trim-o-matic. Then we need to remove this unnatural amplification that is biased towards specific types of read sequences. And so that is called PCR amplification. You can see it's the process of after your genetic material is in the flow cell, there is a phase for bridge amplification before it goes into the sequencer. And that variation could be adjusted by going through the PCR clean step. Afterwards, we're going to map the reads that we have cleaned up onto the transcriptome. So mapping on the transcriptome allows us to identify the start and the end position for each read and create what's called a SAM or a BAM file. So this file will contain the sequences of the reads, as well as the beginning and the end of each read on the reference genome. How many of you have a sense of how long is the human genome? So who knows how long is the human genome? Okay, so NG is saying 3 billion. Please specify 3 billion what? 3 billion pixels, 3 billion meters. What are we talking about here? Nucleotides. Okay, great. So that is almost correct. It's about 3.2 billion nucleotides, so individual letters in the human genome. And so you can imagine how mapping or finding the best position for that read to be aligned to onto the reference genome is a time-consuming application. So here you can see that this will take some time. After we have mapped those individual reads onto the reference genome, we want to quantify them or understand the levels of expression. How much of this particular exon, this particular transcript, or this particular gene do I have in a given sample? So that gene expression quantification step will produce what's called an expression table. And we can measure these levels of expression in several different formats. We can just count how many reads do we have, and so that will be called read count, or we can normalize the read count based on different properties, either how many reads we have in total for each sample, because the numbers could be different, or also include the length of a gene. So if we have a very long gene or a very short gene, the likelihood of that particular read to fall onto that sequence is greater, and so we can account for that as well. Once we have quantified our expression levels, we need to end this pipeline. So just click on end, click OK, and now you will see a download pipeline output file section, which you can open and find here some of the outputs. Okay, so just to get a sense how many of you were able to run this demo pipeline and you see the results. All right, so MHC18 is saying yes. Okay, Francisco. Excellent. Let's give it a moment for all the rest of you that are maybe still preparing the pipeline. It should look something like this. Anyone else? Is anyone else getting results, or are you having any trouble going through this sequence? Okay. All right, well, let's move on. What exactly should I do? So, NG, are you here on this screen? Okay, so click right here on this drop-down arrow and go to this demo RNA-Seq cell line project. Once you click here, you should view the pipeline builder. Do you see it? Okay. Once you see this, you click on Start. Okay. Click on OK. And now you will see some additional options. So the one we want to do is click on Trim-O-Matic, PCR Clean, connected to Bowtie 2T, which is mapping on the transcriptome, and then RSEM expression table. Once you have completed these steps, you click on End, and then you should see this output data right here. And don't worry, we will go through all of these steps, and as you saw, there are many different options as we went through this process of building the pipeline. So we'll discuss some of these options and exactly why do they matter and how do they change the outputs that we see right here. Okay, so just to give you a sense of how this works, right? This will be included in many of your assignments and many of the things that we will do in the interactive sessions. We will go through different pipelines, we'll discuss the different methods, and we'll also talk about the input and the output file so that we understand how do we get from a sample, a biological sample that contains some extracted RNA, all the way to a table of digital data that we can quantify and now work in a statistical way with the numbers that we have to understand how does that characterize the samples that we put into the sequencer. So as I mentioned, there are going to be assignments like this, and there are going to be practical sessions where we will review the assignments. And once we review those assignments, you will be able to submit the sessions. And so in those, you will utilize this T-BioInfo platform. Now, importantly, this platform is really built for biologists and not necessarily for bioinformaticians. It's important because many times when people think bioinformatics, we go to Linux and we start bash scripting from the command line. And not that that's a bad way to do things, it's a good way to do things, but it precludes many biologists and clinicians from working with their own data. And we want to make sure that all of you get access to tools that you can use and actually get some results from different types of data sets. Because unless we try to apply this technique to many different types of data and try many variations of the same pipelines, we will not appreciate the opportunities to explore this data in greater detail and really getting insights into the biology that we are studying from the data that we have. Okay, so in the pipeline, there are many different options. And from those of you that already have access to this platform, what you can do is you can start exploring each one of these variations and see the different types of logical steps with the specific outputs that you will get as a result. And ultimately, we want to understand what the data describes in terms of biology. So we'll go from the basic processing to statistical analysis to biological interpretation. Here you can see a pathway, and this pathway helps us understand how the differentially expressed genes that we found in the data set help explain some kind of a biological pathway that we can see right here. Now, we'll also look at the outputs of intermediate steps. I already mentioned the SAM and the BAM files, so we'll visualize those SAM and the BAM files. We'll look at tables of expression, and we'll understand the variation in those tables of expression, looking at them from different perspectives. Again, we're talking here about 3.2 billion nucleotides, about 25,000 genes, and we'll look at even more complex data sets where those numbers will grow. So it's important to know how to leverage the different types of tools for visualizing and exploring the data to really understand what they mean. Now, in the projects that we will explore, all of these techniques will help you understand how do you go from having access to data or generating your own data to gaining biological insights. And in different sections of the transcriptomics course collection, these are broken down into these specific courses. Transcriptomics 1 will talk about processing. Transcriptomics 2 will help us understand how to find genes that resemble a meaningful variation based on our understanding of our samples. But we will also talk about specific ways of pre-processing the data so that we can explore that variation, knowing the assumptions that some of our methods of analysis will expect from the data. So we'll also talk about different types of data mining and classification methods that allow us to work with large data sets that have maybe tens or even hundreds of samples, explore the variation from without having to go through each individual gene, because the variation is going to be very complex. So we'll talk about examples of projects that have a small number of samples, and that will establish our biological understanding. And then we'll go to larger numbers of samples where we will face this challenge of big data. Importantly, some of these methods are written in R and in Python. So you don't have to just stick to the platform. Once you understand the logic of the analysis and the expected outputs, you want to go behind the scenes and understand how does that actually work and learn a little bit of coding. It's going to be extremely beneficial for any of you that want to actually apply these techniques. And so to help you get started and do that, we have developed a resource called OmicsLogic Code Playground. And we will use that code playground together in the sessions that we will do. So let's go to code.omicslogic.com. Okay, so let's go to code.omicslogic.com. Again, here you should see your account if you have already registered. This is actually a free resource, so you can get started here for free. Anyone that doesn't have an account yet, you can just create your own account with just an email and a password. Once you are in, you will have access to these courses. Okay, so please let me know once you see the courses that I am looking at on my screen. Okay, so just so that I have a sense of how many of you are still there. Okay, so David is saying you're not able to log in following any link. So that's very unfortunate, but the way that you do this is you click on Get Started, and here you can create an account right here. Okay, so for those of you that already have an account, and maybe David, if you are having trouble after this session, we can just connect separately and we will guide you through on how to do this. Okay, so here you have some resources. Again, these resources are built so that they could be incorporated into the training that we have. And so they will be related methods of analysis. You can explore this on your own, just kind of any resource that you want to start with. But specifically, what we will do in this program is going to be reviewed right here. So we'll start from loading data, how to load in a table of expression and understand what kind of variables we're dealing with. We'll go to data visualization, and we'll see how to make bar plots and all kinds of box plots and scatter plots. We'll look at dimensionality reduction, which helps us explore variability in samples based on a table of expression. And we'll see how to do that. And just to give you a sense of how this will work, again, you can take a look at this on your own, but how this will work, we will go through this interactive experience where in a session, we will go through a piece of code, understand what it does. All right, excellent, David. I'm happy to hear that. And then we will go through what the input file is, how do we prepare it for analysis, and then what are the specific functions that we have to do to the data to get the results. And then you can just simply click Run. Okay, and actually here we probably don't need this. Let me see. Okay. Something happened here with, let me see, just, okay, there. So we can run this code right here in the browser. And once you understand this code, you'll be able to modify it so that you understand what are these outputs. So if we look right here on PCA plots, we'll talk about, well, what is the biological or biomedical significance of being able to look at the gene expression data to understand what are the different types of breast cancer that we are looking at right here. Okay, again, this is something that you can start exploring on your own. And as we go through this content in the interactive sessions, we will guide you to specific assignments that once you complete them, again, so here you can see that I have next, and you will be able to complete this lesson, and that will all be tracked under your personal account. So you'll get points for completing some of these exercises. For those of you that are going to apply this to different types of data sets, we will also discuss on how to now modify the code in the session and then take this code with you and open this up in your own application like RStudio or an environment like RStudio. So how to install it, how to get it set up, how to get your working directory set up so that you can run this on the data sets that you have on your own computer. Again, if you have any questions as we're walking through this, please do let me know. Now, importantly, on this playground, we will be able to visualize data. We'll be able to understand what this data variation means in terms of biological significance. And we will also talk about a variety of different applications that are very standard to the process of the RNA-Seq processing, analysis, and visualization. So as we go through this process, you'll find tools that are described as a general method. For example, clustering is a general method, or box plot is a general visualization method, but also specifically, what does it mean in the context of RNA-Seq data analysis? So some specialized methods like differential gene expression analysis or annotation of differentially expressed genes for gene ontology terms and pathway analysis will also be included. So for example, one of the things that we will do is we will go through the pipeline that combines processing and differential gene expression, and then we'll see how to create your volcano plot and even heat maps so that you can not only understand what's in the data, but also present your results to others. As I mentioned, at the end of this process, you will see how processing and analysis of bulk RNA-Seq is changing as we enter into this era of single cell data analysis. Now, single cell RNA-Seq has been really the result of the last decade. You can see that one of the first methods was published in 2009. So we're really talking about less than 10 years or just over 10 years ago. And since then, the number of methods has grown tremendously. So the methods became more and more sophisticated, more and more automated. And today we're looking at a number of established methods that we need to understand how they work and what do they mean. And single cell transcriptomics really is focused on identifying all messenger RNAs that are present in a single cell. Unlike bulk RNA-Seq profiling, where sequencing libraries are generated from thousands of cells, single-cell technologies isolate cells and generate cell-specific sequencing libraries. So we need to know the barcodes for each one of the libraries, which gives us a special technical process to prepare the data and then also produces a table of gene expression that is very different from your bulk RNA-Seq, because you have thousands or maybe hundreds of cells, and many of the genes are just going to be zero because we don't have as much genetic material coming from a single cell as opposed to a bulk data set that we have. So that's where a lot of the methods for preparing the data and then working with the output tables are going to be different, but theoretically and logically, they will be based on similar steps. But we'll go through that process towards the end of the program. Now, when we speak about the application of these different methodologies, we have to understand that, let's say in biomedical discovery, they also help us view the process, the biological process that we are studying at different levels. We can look at patient-derived cell lines where we have a lot of control on how the cells look and how they function together, but really do not represent the systems aspect because they are just individual cells. When we look at patient-derived xenograft models, we are looking at a host, like a mouse, with a grafted-in tumor that comes from a patient. But ultimately, our interest is to understand how can we study these phenotypic variations at the patient population level. And so that's where we will see the connection between different types of data, different types of methodologies to work with these models, and then ultimately how to interpret them using different types of data sets. So we'll look at NCBI, where we can get a lot of patient-derived cell line data and patient-derived xenograft models, and we'll look also at data sets like TCGA, the Cancer Genome Atlas, that really has a well-characterized multi-omics data repository for different types of patient cohorts. And we'll see how access to such data, as well as the development of the specific tools and methods that we will discuss in this program, have really changed different types of applications. For example, breast cancer classification has changed over the past decade tremendously because of these molecular technologies. And we'll discuss how that is happening, and we'll also discuss how we are still not there. We have not cracked the code for some of these diseases, or many of these diseases, in fact, because the biology is so complex. However, we will talk about some of the applications of machine learning for this type of molecular data, which helps us really understand how RNA-Seq and similar technologies are changing the face of precision medicine. So we'll talk about modeling precision medicine. We'll have a data set that we'll work through to identify the relationship between RNA expression and prescription of different therapies based on an example project. In that project, we'll not only learn about the actual technical terms, but we'll also go through some of the terminology that you will encounter in typical publications in this field. And it's important because ultimately you would want to go back to the publications that are being discussed in the media, that are being discussed in the research world, in the academic world, and the industry, and to really understand what each one of these terms means. And so we'll see specifically how all of these methods are, and visual representations of the outputs from these methods are going to become much more clear after you go through this program. Now, I did mention that I will be just one of the presenters. Together with me, we have a team. Dr. Harpreet Kaur and Dr. Mohit Mazumdar will also be involved in some of the sessions, as well as project discussions at the end of this program. So it's important that you can find them. They're here on the chat together with us. They will play different roles as we go through the program, the training program itself. But as you develop a sense of how the RNA-Seq data is analyzed, you'll start thinking about how to apply this data to your own project. And that's really where I see them being most helpful, is helping you determine how to go from training to independent research. And we have multiple opportunities to go through publicly available data sets, to go through some of the research papers that you might be interested in, and discuss how to apply this data to a project of your own. We'll guide you through that project in one of the last sessions that we'll have. But as you start going through the sessions that we discuss here, if you have any questions about that, or you have specific ideas that you would like to discuss, please do reach out. So for Dr. Harpreet Kaur, her email is support@pine.bio. You can also email her any of the questions that you might have at the technical level. And for Dr. Mohit Mazumdar, who also has a PhD in computational biology, his email is mohit@pine.bio. Thank you, Bipsa. So if you have any questions related to different types of programs that we have, which includes a research fellowship that you can join to work on an independent project, or you have questions about how to find and organize your data for independent analysis, you can reach out to him. And any technical materials, access to different types of accounts, different platforms that we discussed today, you can reach out to Bipsa Biswas. Her email is marketing@pine.bio, and she will be sending you a lot of the information about this program and about the different resources that we have prepared. So please put her in your contact list so that you don't miss out on those important emails. She will be also emailing you about some of the recordings, or not some, but all of the recordings that we have for these sessions. So after the session is over, you will have to wait a little bit, and then you'll get a link by email and also on the forum about the recorded version. Now, all of our team has gone through the same training like we are discussing here. And so we are familiar with the materials and the different assignments and the quiz questions. So please feel free to reach out to us. And we hope that as a result of this program, you will also feel more empowered and able to analyze your own data. And we are looking forward to working with you to identify how we can best support that process from our side. Now, briefly, let me pause here for any questions. If you have any questions, put them in the chat box. And then I want to talk a little bit about the different projects that we will cover in this session.
Hi, Ilya. If I may say and address a few things. So I know that I have sent all the registered participants an email on all the resources that Ilya has discussed today. Now, one thing that I wanted to point out is that we have 3 different platforms, as you see. One is the educational platform for the different asynchronous courses, and there are also asynchronous projects, and the forum. And the second is the T-BioInfo server platform. And the third is the OmicsLogic platform. Now, all of you must have received an email on T-BioInfo access from me that is only for login to the T-BioInfo platform and is not applicable for the Edu platform or the OmicsLogic platform. And then you will be able to use your single email, whatever email you have registered for in the program, for the other 2 sites, which is the Edu platform, which most of you have access to, as well as for the OmicsLogic. If you're not able to log into the OmicsLogic, please create a new account for yourself. And in case you have any problem, please reach out to me again, and I will be able to help you out with that process. And again, thank you so much for attending today's session. And Ilya, back to you.
Okay, thank you, Bipsa. Okay, so before we actually continue to talk about projects, let us briefly ask you to also join our community. So as I mentioned, this is a program to help you learn and master RNA-Seq. We have other programs that include non-training programs, include free resources, as well as our research. So we ask that you also join us on our OmicsLogic group on LinkedIn. If you have a LinkedIn account or a Facebook account, Bipsa, if you could please paste those links. Okay, so you will find there a lot of the resources. Okay, let me just go there real quick. Okay, thank you. Yeah, so we have here the OmicsLogic group where we have over 700 members, and we regularly post here some of the results of this training. So you'll see, for example, student projects that have joined the training programs without any background or experience in bioinformatics and have been able to develop really impressive research projects using some of the tools and the methods that we discussed. So really looking forward to interacting with you, not only in the program, but also beyond. Okay, there was a question if there are some free courses with certificates. Dr. Kumar Singh is asking. So Dr. Kumar, we do have some free resources that you will get certificates for. And also for anyone, we do ask that you help us understand a little bit more about you. And the best way to do this is to finish our Introduction to Bioinformatics course. The Introduction to Bioinformatics course is free for anyone to take. It also includes introductions to some of the methodologies that we will cover, but importantly, it will guide you through a survey where you will be able to share a little bit more details about yourself. And we're looking forward to understanding more about your background level, your interests, as well as where you are located. So please start with this free course, and you will find some of the other courses on this educational platform. You also will find some materials for coding that are free on the code omicslogic.com, and all of that material together is going to be available on our page called learn.omicslogic.com, which you could also explore by going here. The same account is going to work here as well. Okay, so let's briefly summarize what we have talked about and talk a little bit about the different projects that you can learn about these different methods and their applications by going through these projects. There are many different applications of RNA-Seq, including a lot of the different cancer projects, including neurodegenerative diseases and even agriculture. So we do ask that you review some of these materials and think about how you can go through some of this type of data on your own. Now, importantly, some of you are asking about the research fellowship. The research fellowship is a program that will help you go beyond training and apply the tools to a data set of your own. In this program, it is a 3 or a 6 month research fellowship program. It is not free, but there are scholarships available if you have experience and you already have a defined project. You can apply for a scholarship. The scholarship also applies for the training as well. So Bipse, if you could share the link to the scholarship request form. Now, the research fellowship, after you've understood how RNA-Seq works, you might want to develop a project of your own, and that will take significant time. So 3 to 6 months is typically what it takes, depending on the complexity and how prepared you are already for the coursework. Bipsa, if you could please, thank you. Well, no, if you could post the request for scholarship form. Okay, so previously we have numerous participants that have completed the training, went on to develop a project of your own, and have presented those at different types of international workshops and poster sessions. Now in the days of the pandemic, this is especially meaningful because so many of these are happening online. So it's an opportunity for you to learn more about these international sessions as well, and really develop something that is impactful. We also hope that some of the materials that we discuss in these sessions go beyond just learning and kind of practice projects, but as you can see, different researchers have joined these programs to also apply the same approaches to clinical impactful projects, and we hope that we can facilitate that as well for some of you. So if we know more about you, we know more about how to help you, we can guide you to the resources that will be most meaningful to your personal development, right? So whether it's a career path, whether you are interested in training your students and helping your students understand how to utilize RNA-Seq data, if it is development of a research project, or even applying that research to a real research problem. So we have different resources and tools that are available for that as well. So in summary, in this program, you'll learn about RNA-Seq data processing, analysis, and interpretation. We'll leverage cloud-based tools that will allow you to go quickly through some of the complex pipelines that are typically used in RNA-Seq experiments. You'll see where machine learning fits in to this large biomedical data set analysis paradigm. And you'll be able to complete and present an independent research project if you have enough time, and obtain a certificate of completion or certificate of excellence, and you'll be able to showcase that on your social media profiles, etc. So we are excited about this program that is starting. If you have any questions, please follow up with us by email or on the forum if you're already registered. And in our next session, we'll start talking about the process of preparation of the data and data processing. What are some of the options that we saw in today's pipeline? And what are some of the differences and outputs that we expect from different processing pipelines? Again, my name is Ilya Brodsky. I'll be here in all of the sessions or most of the sessions, but also thank you, Bipsa, for joining today and helping with the session. And Dr. Harpreet or Mohit, if you have anything to add, feel free to add now.
Yeah, hello everyone. Myself, Dr. Mohit, and I'll be happy to help you with this process of learning, transitioning from hands-on exercises and theoretical aspects to a research project. So please feel free to reach out to me. Happy to help. Thank you.
Thank you, Mohit. Harpreet, would you like to say a few words?
Yeah. Hello everyone. My name is Harpreet. So I'm going to take you some of the assist you during all those sessions. So even if you face any issue regarding any course or any assignment during all those courses, transcriptomics courses, so you can mail me at support@PineBio, as already Ilya has given that you to in the chat box. So you are feel free, please feel free to regarding any query if you face any issue regarding any courses or the assignments. Okay, so thank you everyone.
All right, thank you, Dr. Harpreet Kaur. And we'll see all of you in our next session on Friday, December 11th, at the same time. So these session links will be emailed to you, and you'll be able to also see the details of what is the topic of the next session on the forum. The feedback link, Dr. Kumar Singh is asking for a feedback link. I don't know, Bipsa, if you have one, please share it with him as well. Thank you everyone for joining, and we'll look forward to seeing you again in our next session.